So now we will discuss preparation and hybridization process of fish in detail. First of all, we'll discuss preparation of probe, how and what type of probe they are used. Probes may be labeled with fluorescent or radioactive labeled, and these fluorescent labels, they're generally preferred because of the risk associated with radioactive labels. We all know that radioactivity results in different types of cancer and other diseases. That's why radioactively labeled probes, they are not used, and fluorescently labeled probes, they are preferred. Length of probe, they may vary from few bases to 1 million base pairs, and probe size is important because longer probes, they are hybridized less specifically than the shorter ones. So we should try to use smaller probes as compared with the lengthy probes. The reason is longer probes, they will not bind specifically, they may bind unspecifically as well. Signal strength, that depends on probe labeling efficiency. So how a probe is labeled, type of probe which we use, whether it is small, large, whether it is fluorescent labeled or radioactively labeled, and similarly the type of the dye which we use. So जो probe के साथ जो डाई है, क्या वो सिंपल डाई है या बहुत अच्छी किस्म की डाई है, तो इसके ऊपर भी सिग्नल जो स्ट्रेंथ होती है वो डिपेंड करती है. आमतौर पे जो एलेक्सा फ्लो डाइज होती हैं उनका सिग्नल बहुत अच्छा होता है. सो एलेक्सा फ्लो डाइज जो है वो यूज़ की जाती हैं. Probes which are used, they are generally are of three types. So locus specific probes, centromeric probes, and third are the whole chromosome probes. We will discuss these one by one. Locus specific probes, as the name indicates, they are used to identify certain part of the chromosome. I mean, certain gene is identified. So this locus specific genes, locus specific probes are used to locate particular gene in the chromosome. Then centromeric probes, as the name indicates, that probe will hybridize with centromeric position of the DNA. So that's why these type of probes used to find out whether an individual has the correct number of the chromosome or not. जाहिर है अगर कोई क्रोमोसोम मिसिंग होगा तो सेंट्रोमेरिक वहाँ पे विजिबल नहीं होगा, तो उसकी बेस के ऊपर हम देख पाएंगे कि क्रोमोसोम नंबर पूरे हैं या कोई कम है या ज़्यादा है. So centromeric probes were used to find out the number of the chromosomes present in the sample. Whole chromosome probes, as the name indicates, they are used to probe complete chromosome. So what is the procedure? We use many smaller probes labeled with different types of labels, and these smaller or these many probes, they bind with their specific sequence on the chromosome, and in this way the whole of the chromosome is labeled from these smaller probes, and we are able to see the complete chromosome in its distinct color. And this technique where we use we are able to see whole of the chromosome. This is known as spectral karyotype. And now we will discuss hybridization process. So we will see how this probe is bound with the DNA molecule present in the sample. Sample with chromosomes from dividing or non-dividing cells is selected. So generally, dividing or non-dividing cells, they are selected. Chromosomes, they are firmly attached to a glass slide so that they may be able to study later on. Sample and probe is treated so that these two strands, they are separated from each other. Either they are heated or they are treated with some alkali so that two strands of DNA and DNA of sample and two strands of probe, they separate from each other. And then probe is hybridized with the sample for about 12 hours and or even longer time, so that this probe can bind with the sample. And this is followed by washing, so that unbound or partially bound probe, they are separated so that we don't get false positive results. And these results are then visualized by using a suitable detection system like fluorescent microscope or some other types of detection system.